Thanks for coming in and attending this seminar today. My name's Brielle, and I'm one of the organizers of this year's Quantitative Seminar Series for the Converse Lab. And I'll begin by acknowledging the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. We also acknowledge that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. So today I'm very excited to introduce our speaker today, Scott Hotaling. He is a postdoc in the School of Biological Sciences at Washington State University. He received his PhD in 2017 from the University of Kentucky, advised by David Weisrock, and has a BS from North Carolina State University. And in 2015, he co-founded the Teton Alpine Stream Research Project, which is dedicated to using long-term ecological monitoring to quantify how mountain stream ecosystems are changing. And beyond studying mountain ecosystems, he thinks a lot about scientific writing and wrote a paper about that last year that I'll put in the chat. And he's a big fan of Twitter and loves getting out and mountain biking in the Pacific Northwest. So you can take it away, Scott.
Cool.
Thanks again.
Well, thanks, Brielle. It's really exciting to be here. I lost my video when I went to full screen, so I'm truly talking into the void, as is the reality of these situations. So I'll do my best to kind of stay on track and not get lost in my head staring at my screen. But yeah, it's really great to chat with all of you and tell you about the work that I've been doing for the last decade or so, exploring alpine biodiversity and mountain streams across the Northwest. And I also really appreciate the land acknowledgement that you all did. It's something that I'm trying to make a habit of thinking more about and acknowledging whenever I write papers and work on this stuff. I'm lucky to work in a lot of really amazing places that people have lived in and stewarded for a long time before I showed up. And just to highlight what Brielle said, I also do use Twitter a lot. So if you're excited about any of this work or want to connect about it or anything, one of the best ways to find me is at MTN Science on Twitter. So diving into the talk, this is one of my study sites in Grand Teton National Park. It's the outflow of the Middle Teton Glacier. And I think it fully exemplifies how harsh of a place these environments can be for both researchers and the organisms that live there. And so for those of you that don't know who I am, I thought I would give just a really brief overview of how I think about my research in this kind of integrative context of high elevation ecosystems. So I work a fair amount on purely genomics. I work a bit on ecology. I work a bit on physiology. I have explored more of the ecological and genomic interface. And then whenever possible, I wind up in the intermediate point where I'm linking all of those disciplines, but that is a pretty challenging thing to do. And so for this group, I don't think I have to really belabor this point, but climate change is dramatically altering our planet. And so this is a really great infographic by Zach Labe. He has some great stuff that he puts on Twitter, looking at sea ice change over the past like 30 or 40 years in the Arctic. And as you can see, it's dwindling. And that recession of the cryosphere isn't just happening at high latitudes, it's also happening at high altitudes. And it's perhaps most notable at lower latitudes where in temperate areas like the Pacific Northwest, where we have high mountain glaciers that are going away really, really quickly. And so a place that I've spent a huge amount of my time, and I'll talk about more today, is Glacier National Park in northwestern Montana. So here's a repeat photograph from the Boulder Glacier, which is one of the largest glaciers in the park 100 years ago. So you can actually see some people standing on the glacier. And if people can't see my cursor, maybe someone let me know, but I'll assume that you can. So these are people standing on the glacier, and you can actually see a little stream kind of down here below the ice that then goes back under the ice. And there's Thunderbird Peak in the background. And within just 60 years, the Boulder Glacier was essentially gone. And now I think it's considered something like 98% receded. as of 2015 or so. So there still is a tiny little bit of ice, but you can imagine that, not on a millennial scale, but on a decadal, human lifetime scale, this glacier has completely gone away. And for organisms living in this stream, there's still a meltwater stream there. You can go up and there's plenty of stuff living in it, including the stoneflies that I'll talk a lot about today. But you can imagine that that's a massive shift in the environmental conditions, which has all kinds of ramifications for biotic interactions, stress physiology, etc. And so one of the major questions of my work is essentially, can these populations respond to that significant of a change? So how imperiled are they under climate change? We assume a lot based on their distributions. So we assume that they live in these cold meltwater places because they're ultra sensitive to warming and they're the canaries in the coal mine of climate change. But as scientists, we need to actually figure out if that's true and do the best we can to understand that, because that will inform the degree to which we can predict their vulnerabilities into the future. And so mountains in general have long been identified as biodiversity hotspots. And so this map is kind of based on the zonation of mountains that von Humboldt laid out in the 1800s. And a big component of that is heterogeneity, which I'll talk a little more about for streams in a second. But when you think about mountains and their biodiversity, this is a really nice review from a couple of years ago. Essentially, more species are concentrated in mountain ecosystems. So there's less overall land area in mountains, but more species are either only in those areas or have the bulk of their range is within those areas across mammals, birds, amphibians. And if you took invertebrates, you'd likely see a similar pattern across whatever group you looked at. And a big part of that is that zonation. The topography creates more habitat within a smaller area. But historically, people have generally focused on the areas that are the most biodiverse within those systems, and that's areas below, the actual snowy peaks and meltwater right up next to them and the glaciers. And so I was part of a paper that we published a couple of years ago that just essentially, I'll give you the full background. It was a response to a really, really nice paper that detailed biodiversity risks under climate change for glacier-associated species. And they looked at everything from streams and lakes and oceans and 4 fields of glaciers, and they really had this integrative perspective on how loss of a glacier will impact biodiversity. And so we, not trying to critique the paper because we all really liked it, but as a group of glacier biologists, our basic response was like, yeah, all of that is threatened, but the actual biodiversity of the glacier itself and the really, really nearby ecosystems should also be considered. So the title of the paper is essentially the main point of the paper. And that's also kind of the area that I work the most, this high mountain ecosystems. And so to give you all just some special attention for the Seattle-based UW folks, I'm going to show you just a little brief vignette of glacier biodiversity on the mountain. And I'm sure many of you know what I'm talking about. For those of you that don't, I'm referring to Mount Rainier, the big stratovolcano that you can see from downtown Seattle. And all of this biodiversity has been documented on a single glacier on the south side of the mountain. This is all from the Paradise Glacier at just above Paradise, the visitor center there. So biodiversity of glaciers is microbial. It's dominated by microbes. They do tons of biogeochemical cycling, and they're just really important to the overall glacier and ice ecosystem. And for most people that know a little bit about glacier biodiversity, they know that much. There are plenty of people that think there is no life on glaciers, that they are just ecological dead zones. And in my opinion, and in reality, that could not be further from the truth. But it's not just microbes and snow algae and bacteria. There are also these little critters called ice worms, which only occur on mountain glaciers in Western North America. There is a Tibetan ice worm, but I won't talk about it today. But they're an annelid. They're extremely interesting from all kinds of perspectives. They're the largest organism on Earth that spends its whole life cycle in ice. They're really UV resistant. But despite being an ice worm, our physiological work on these creatures suggests they can't actually freeze. So if the temperature, they live at 0 degrees Celsius, but if temperature gets to negative 0.5 degrees Celsius, they essentially freeze to death, which is a very fascinating physiological reality. Just to show you a little bit what they look like, here is an ice worm actually out sensing its world. I call it a terrestrial species, but really it lives in the ice. And glaciers, I don't really know how to classify them because they are flowing water. They're just frozen flowing water. So sometimes I consider them streams as well, just really, really slow streams. And then there are birds like the gray-crowned rosy finch, the highest elevation nesting bird in North America. And rosy finches actually feed on ice worms, and we think that they actually use ice worms to support a really key reproductive period in their life history in kind of early June when they're laying eggs and brooding their young. And then continuing up the trophic cascade, we've documented American kestrels chasing around and hunting species like rosy finches on the glacier. And this all kind of makes a trophic cascade of, we know ice worms eat microbes and snow algae. We know rosy finches eat ice worms. We know kestrels appear to hunt. We've never actually documented them killing a songbird, but we've definitely seen them actively hunting them on the ice, which makes feeding on ice worms, you know, being a dark bird facing the ground on a white background, a really dangerous game. And we have a little bit of evidence that suggests the birds actually preferentially feed in the margins of the glacier, we think, to kind of have some safety and a more camouflaged background of the rocks, like you can see where the kestrel is here. And then a really exciting thing from last summer was we actually got wolverines on our wildlife cameras up on the Paradise Glacier, which, for those of you that have followed the press about wolverines in the South Cascades, this is one of the only documented wolverine families in the last 100 years in the area. And so they could be feeding on things like, not necessarily kestrels, but we know they hunt marmots and other species. But we aren't here to talk about terrestrial biodiversity, so I just wanted to give you that vignette as a slight educational detour. We're here to talk about aquatic biodiversity, and the star of this show will be stoneflies. These high elevation stoneflies, often in the genus Lednia. This is Lednia tumana from Glacier National Park, that I study using them essentially as a model for an entire community of organisms that live in the meltwater of glaciers and snowfields that might be at risk under climate change. So in 2019, Lednia tumana was listed under the U.S. Endangered Species Act due to climate change-induced habitat loss because of its distribution. So here's a really amazing little video by my colleague Joe Gersh showing Lednia tumana in its stream. It's a little choppier than normally would be because of, I think, the connection. But this is a caddisfly called Rhyocophila ebria that we see in a lot of these places. Here's more Lednia. And then in a moment, we'll actually see a mayfly. So here's a mayfly over here. And the point of this is to not only show you something pretty, but to also highlight that Lednia exists as part of this broader alpine stream invertebrate community. And so protecting it or thinking about risks to it also will inform a lot of other species conservation. And so mountain heterogeneity also drives stream biodiversity. And the way that essentially works is you have a lot of different hydrological sources in these habitats. So you have, like, this is a volcano in Ecuador. You have a glacier-fed stream here, which is very cold and very turbid and has really a highly variable flow based on melt rate of the snow that feeds it. And then, like, just next to it, coming into it and merging, is a groundwater-fed spring, which has some amount of some version of an aquifer and is much more stable in its flow, warmer, etc. And then you have streams like we have in the Teton range, which this is actually the same stream as my cover photo of this talk on a much nicer day. And this is fed by the outflow of the Middle Teton Glacier. And here it's hard to even tell where the stream channel is. It is a stream that is consistently there, at least so far in our project, but it's extremely harsh. It's very turbid. You know, whereas over here, you could potentially have fish getting into some of these streams. I mean, they're at least accessible. You're not going to get any fish up here. And so You just have different kind of trophic ecology beyond just the invertebrates that dominate. And then there are streams like this one, which is pretty exciting for a lot of our work, because this little talus field over here is actually the toe of a rock glacier. And so this is ice melt that's bubbling out of the ground. And it looks like a groundwater fed spring, but that water is at like 0.5 degrees Celsius. So it's extremely cold, like a glacier fed stream, but then it's also clear and stable and has all these characteristics of a spring. So you have this kind of mashup of different ecological characteristics, which then drives a greater amount of regional biodiversity in these habitats. And so the basic prediction for how climate change will impact mountain streams is that loss of glaciers will mean loss of species, largely because you're losing one of those habitat types and the potential specialist communities that live within it. These are data from a large-scale study by Dean Jacobson and colleagues back in 2012, looking at this pattern using the space for time framework in Alaska, Europe, and Ecuador, and they found the same pattern throughout. And so as we look towards the future for mountain streams, we have a basic research and conservation challenge. So the first thing is that we lack basic understanding of high elevation biodiversity, including simply what species live there. But then for what actually lives there, we don't understand the ecology of these species very well, who eats who, what their resources are, what limits their distributions. We don't understand their physiology, and we don't understand their evolution of how did they get where they are, which can be a really useful thing to know when you're thinking about where they're possibly going. And so here are a couple of those stoneflies again, Lednia and Zapata Glacier, both of which have been classified as endangered under the ESA for the loss of glaciers and snowfields. And we need more robust tests of predicted biodiversity loss in space and time. And so, for instance, that Jacobson et al. Study is fantastic, but it was only a space for time study where they took a bunch of different streams with varying amounts of glacier cover and they compared the communities against one another. I think we actually need robust tests through time, most importantly, so we can actually track how an ecosystem is changing and then see what happens. That is obviously really challenging because there's the old adage, the best time to plant a tree was 20 years ago. We don't have long-term data, but we can go out and survey space for time. But we should recognize the inherent noise in those data sets and just be mindful of that. And whenever possible, begin monitoring programs so we have better data in the future. And then there's a really pressing need to link underlying genotypes and particularly phenotypes to predicted threats. Because as it stands, we don't know what factors actually threaten these species. Based on essentially the Endangered Species Act listing and other data centers around distributions. And while distributions can be really informative about what an organism can survive, they don't necessarily tell you where that organism would like to be or where it responds best, because biotic interactions that are independent of temperature or glaciation or whatever could be driving where a species like Lednia tumana exists today. And so in 2020, we kind of laid out this complex challenge of present day conditions in mountain streams and what they might look like as soon as the end of the 21st century, depending on where you are with rising temperatures, declining flows, increased UV stress, and all these various factors which will interact to dictate at least the physiological conditions of what can live in the streams, which then might cascade to inform the biotic conditions and what biotic relationships are existing, who eats who eats what, that kind of thing. And so to move into the more empirical component of this talk, I want to reorient you back to these three species. When I talk about individual stoneflies, I'll be referring to these in the next couple of studies for the most part. And here is a paper that we published 5 years ago or so, just to give you a sense of what these communities look like to some degree. So here is a stream in Glacier National Park coming out of the Sexton Glacier. Red circles are where we sampled in this stream. And for Lednia tumana, the red number is the proportion of the total community that Lednia tumana comprises at that site, and the number of taxa overall at each site. So as you can see, as you go downhill, you get a lot more taxa, but Lednia comprises 40% of the overall community up at the headwater, so it's Lednia and chironomids are the dominant taxa, but that falls off really dramatically as you move down the stream. And then I also want to highlight here, this is temperature on the y-axis in degrees Celsius. And so like this upper site where Lednia is one of the most dominant members, it's the most common organism at that site, I know that. It's essentially like 0 degrees. And these are summer temperatures. So it's getting meltwater straight out of the glacier. And then once you get down, even a kilometer from the glacier, the maximum temperatures, like this little gray area in summer, is something like 7 degrees. So these are pretty cold streams, but the gradient becomes really exceptional as you get closer to the ice mass, which is, not surprising. So this brings us to the kind of overarching question for all of my research in these habitats. That is, are entire meltwater communities at risk under climate change, or are they as imperiled by glacier loss as we think? And so the basic prediction is, using a space for time model, when glaciers recede, species will be locally extirpated. And on regional scales, this means extinction of endemic species like Lednia tumana. And so we have the first study I want to tell you about is a pretty Herculean test of this idea in Glacier National Park, Montana, that took us about, it started before I even started my PhD, and then we published it last year. So it was something like a 10 to 12 year effort. And so we went out, and this is a map of Glacier National Park. Every circle is one of our study sites across the 1 million acre park. And we quantitatively sampled invertebrates from 129 sites in 36 catchments, working with our USGS glaciology partners. We mapped all of the present day ice in the park, both glaciers and any snowfield, anything like that. We calculated glacier cover change since the Little Ice Age, so essentially contemporary glacier recession, using moraines and other data that I will leave to the glaciologists to understand. But we essentially had a 170-year record of glaciation across all of our sites, and we could then rank them as sites that have lost 100% of their ice, 20%, 0%, whatever, across a bunch of different basins. And here's an example. Red dots are where we sampled. Yellow is the Little Ice Age margin of the glacier. And then present day ice you can see in white. And all of these areas have receded really dramatically over the last 170 years, or even just contemporarily. There's the little tiny Boulder Glacier I talked about earlier. And you can see a little bit of ice that's still there. And that was in 2009. Like, I've been up there since then plenty of times, and I do not think there is still that much ice up there. I would say it's something like a quarter of that. And so looking at the high level patterns, some just parts we wanted to make sure that what we were seeing made sense in our analyses. We did see that species richness increases as you get further away from, or you get to areas with less glacier cover. So high glacier cover over here, lower glacier cover over here. And then beta diversity, so how similar communities are to one another, also decreased as you got to areas with lower glaciation, which has been seen previously. So that was a nice kind of confirmation of what we were seeing. Here's a figure from a kind of the first paper to show this relationship in 2007 by Lee Brown and colleagues. And if we flip Lee's plot, we get, to make the axes match, we get essentially the same pattern. And so we used this approach called latent deer sleigh allocation to ask what communities exist in Glacier National Park in these headwater streams. And we based this on we incorporated, I should have said this on an earlier slide, a temperature model of the entire park, which had been published previously, which was based on a number of temperature loggers that were deployed around the park for a couple of years. And we identified this rare cold water community that appears to exist. And so when we split all of the sampling into two communities, we can identify species that are abundant in cold water, so things like Lednia tumana, Rhyocophila ebria I mentioned earlier, and then species we know are more generalist thermally, and they're in much warmer places. One that I've studied a lot is this warmer water caddisfly that sometimes in mountain streams called Rhyocophila bellona. And then we could see which ones were like most strongly associated with these cold water habitats and stuff. Was one of those was Stegobromus glacialis, a little high elevation amphipod that we see every once in a while in the park. And when we looked at the relative abundance of that cold water community relative to the more generalist warmer water community, we found a noisy pattern, but a pattern that as you moved from high glacial cover to low glacial cover, we tended to see less abundance of that cold water community versus the other community. But I want to point out here that we actually also had some streams in our data set, something like 50, that had no present day ice feeding them, surface ice feeding them. And here we would have expected, I would have predicted that we would see essentially no signature of that cold water community in these areas that have no surface ice. But that wasn't the case. We had some areas where there was a pretty high proportion was this cold water community. Now, if we took those same sites and looked at what happened to that community as we moved further from the source downstream, we saw that for those no glacier sites, there was a really rapid decay from the cold water community to the more generalist community as you move downstream. Like by a kilometer, you'd lost basically all of it. But in the high glaciality, or in the glacial streams, that pattern was much less decayed. There was this kind of buffering where the glacial meltwater was allowing the cold water community to persist further downstream. And so circling back to imagining what this all means, it appears this cold water biodiversity can persist in habitats after glaciers recede. So this is just the 50 sites that had no glaciers since the Little Ice Age, green, or that had lost their glaciers over the last 170 years. So similar but slightly different categories. And so for no glaciers, so no ice in 170 years, we see that a bunch of the sites have none of that cold water community abundance. So 40% have none, but a big proportion have a lot. And then if we look at the ones that lost their glaciers sometime in that last 170 years, we are not able to date when glaciers were lost from an individual catchment over that time. We see a similar pattern. And we can take that to a bit more of a simplistic view and just ask, how many taxa are in all of the streams, and how many taxa are present in streams that don't have ice? And there are 112 in all of the streams in the study, and 110 in sites that don't have ice, which was a pretty shocking number for us to kind of wrestle with what was going on here. So this somewhat contradicts the prevailing theory that there's going to be this process of turnover in these headwater streams, whereas climate change precedes, warmer water, lower elevation communities will shift uphill and displace this kind of cold water specialist community depicted here in blue. And so we would expect under that scenario, you would get something like this, where as you move through time, local richness at an individual site will increase, but regional richness will decline as those cold water species, these blue circles, blink out on the landscape. But our data supports something more akin to what we call a tolerance model, where you have the same starting point at the Little Ice Age, perhaps, but then through time, these lower elevation species shift into the higher elevations, and you don't actually lose those cold water species, but you gain the lower elevation species. And so your regional richness stays essentially flat while your local richness increases. Now, obviously, there are a lot of things that we could be missing here. There could have been recolonization of some of these habitats by other species like since sometime in the Little Ice Age. There could be some kind of lag effect where if we checked back in 100 years, we would actually see more of that turnover model. and we just happen to catch it at an earlier time point. But whatever is going on here, it's intriguing. And the last thing I want to point out is that one thing that I think could be going on, which I'll circle back to in a little bit, is the idea of subterranean ice. So things like that rock glacier maintaining cold water conditions despite not looking like they would in a basin based on satellite imagery of ice. And that's a really exciting idea for a lot of reasons, which I'll talk about more in a minute. And so that is the community ecology perspective on what's going on in these high mountain systems, and this idea that these species might be more resistant to climate change than we may initially guess based on their distributions and some space for time approaches in other mountain ranges. But I wanted to take a more mechanistic view to ask if these cold adapted stoneflies can actually tolerate warming. And when I say I wanted to, I need to clarify that to we wanted to. And this is work that I've done in lockstep collaboration with Alicia Shaw, who is a new faculty member starting at Michigan State this year, and who is an exceptional invertebrate physiologist working on aquatic insects and all kinds of related questions. And so Alicia and I paired up to ask first about the thermal tolerance of these mountain stoneflies, predicting that they can't tolerate temperatures far beyond those they naturally experience. That would be suggestive of they live there because they're really sensitive. They're the canaries in the coal mine. And then also we want to take that to the level of gene expression and actually look at what their cellular response is to heat stress. And so here we expected they would have a strong cellular response when at these kind of maximal temperatures they can survive, and that we would see that in genes like heat shock proteins, which despite their name, they're really just stress response chaperone proteins, which are expressed and produced when a cell is under stress, whether that's cold stress, heat stress, some kind of pollution, UV, whatever. So it's kind of two-pronged experiment. And so Alicia and I went out and we sampled stoneflies from 6 different streams, one in Glacier National Park and 5 in Grand Teton National Park. And we sampled those 3 species I discussed earlier: Lednia tumana from Glacier, Lednia tetonica from the Teton Range, and then the species Aquatic Glacier. And so just to show you what the temperature variability looked like in these streams, this is just a 24-hour typical summer day. And as you can see, sites like Cloudvale Dome, which is right below a glacier, they are essentially flatlined during the day and they don't change very much and they're pretty cold. But then sites like Lunch Creek experience this pretty strong afternoon spike in temperature. And so we wanted to have a range of temperature in the sites because we thought that might predict thermal tolerance. And so how did we measure thermal tolerance? We used CT max, which is a temperature when locomotion becomes disorganized, the critical thermal maximum. And we measured this as the riding response. And important here, this is survivable. And so on the left-hand side is normal Lednia behavior. We would turkey baster them, spit them out, and the little nymph just runs around and it's perfectly happy. And we're slowly ramping the temperature during this experiment. And on the right, here's an example of loss of riding response at the CT max. And here you can see a stonefly that is essentially, it is alive. So when you put it back at a lower temperature, it recovers quickly, but it no longer is able to organize its locomotion. And so CT max varied predictably with thermal regime. So CT max on the y-axis here, T max of the stream is on the x-axis. And as you can see, across all of the species in the study, we see the same kind of relationship, and it's even stronger within the 4 Lednia tetonica populations. And notably, I want to highlight, look at this CT max. Like, these things are tolerating 27, 28, 30 degrees Celsius when they never see more than 15 in the wild. So much, much higher temperatures in the short term than they would naturally experience. So that was already pretty interesting. So we went in and we did some RNA-seq on 3 of these populations. And we first just asked, what's going on with gene expression globally? So in looking across, I think the 5,000 most expressed genes, we saw some interesting patterns that there was some split between the two species, which we might expect. There wasn't a very clear treatment versus control globally pattern in Lednia-Tumana, which I won't squint too much at this or make a big deal about it, but I think it is worth noting that Lednia-Tumana tends to experience much warmer streams and much more variable streams than Tetonica, at least as far as we can tell so far. And Lednia is more broadly distributed on the landscape, or Lednia-Tumana. So it does make us wonder if Lednia-Tumana might be just inherently more able to deal with temperature stress than Tetonica. But in Tetonica, we saw this really clear treatment shift across the two sites we were studying. And so if we take that and look at the most differentially expressed genes, the ones that are significantly up or down regulated, there were something like 60. And for the purpose of this, we go into much more detail in the paper when we have 8,000 words to talk about it, but I won't do that here. I'll just highlight that some of those are things like heat shock proteins, which is what I was discussing earlier. And so to answer the question about, are they exhibiting a strong cellular heat stress response, we went through and pulled out all of the heat shock proteins that we identified in the data set. There were, I believe, 13. I'm only showing 12 here for space constraints. The 13th matched the same pattern. But so for each of these, this is what the data will look like. This is the gene expression in counts per million on the y-axis, and then heat stress, that treatment are the dark individuals, and the lighter circles are the controls that were held at 3 degrees Celsius. And then Lednia-Tumana is in red, Tetonica is in blue. And so for most heat shock proteins, we saw this same exact pattern of constitutive expression, where the heat shock protein is expressed at a certain level seemingly all the time, both at low temperatures and at high temperatures. And so when we look at all of them, we only see a couple that exhibit that clear upregulation pattern that we would have expected. So we concluded from this that we can't really say there's a strong cellular stress response, at least using heat shock proteins as a panel. And this raised kind of a different question. Constitutive expression of heat shock proteins has been seen in Antarctic fishes that are in really cold water. And the idea is that they just never experience heat stress, so they just always turn on their heat shock proteins, possibly to deal with stuff like oxidative stress. So they live in a really highly oxygenated environment, which is also true of alpine streams. But then there's also there's the idea that cold conditions are actually stressful too. What we might be seeing is we put them at warm, they're stressed, but they're also stressed in their kind of native temperature range of 3 degrees Celsius, which would indicate that actually there's some other reason that they're living in those places because it's not their physiological optimum. What we could have done, looking back, is had an intermediate temperature point that maybe would have shown a U-shape to this pattern of upregulation at low temperature and high temperature, but a lower level at 13 degrees C, which then that would have told us that both are stressful. But with our data right here, we can't differentiate between constitutive expression all the time or this kind of two ends of the spectrum both being stressful idea. So we did a little experiment just to test this idea of how is cold stress actually a factor here as well. And we took a bunch of Lednia-Tumana nymphs and put them in a fridge or in different incubators for, I think it was a 1-hour treatment to test how well they could handle a 5 degree C control, near freezing temperatures, or freezing temperatures. And as you can see, there's this decline with a really sharp fall off of 100% lethality at negative 0.5 degrees. So this does indicate at least a little bit of support for, in the summer, for these nymphs, there is some degree of stress related to these kind of more exceptional cold temperatures, as well as these really high warm temperatures. And so now we are actually testing this in a more serious way. And so we've done this experiment but it's in prep and it's being led by Alicia. So I don't want to spoil anything, nor can I yet, because we're kind of trying to get a handle on what this experiment means. But what we did was we went out and we got a bunch of different streams in 2 types. So they are cold and consistent streams, 3 of them, and warm and variable streams. Warm here is really relative, given that 10 degrees is generally considered quite cool. But for these species, 10 is some of the warmer temperatures they will ever experience if they ever get that warm. And then we held them in incubators that were ranging from 2 degrees all the way to close to their CT max. Some of these numbers are a little different in the actual experiment. I made this figure before we had conducted it, but the basic premise is the same. And we held them for a month, and we measured a lot of things: their survival across that time, the timing of their emergence, how well they grew, how they developed their wings as an indicator of their flight performance potential. And then most excitingly, we actually looked at flight performance, which didn't work out super well, but we're very proud of how close we got, considering flight performance is a you could argue is a really strong fitness related trait. Certainly they breed as adults when they emerge from the stream. And it also encapsulates, developmental effects as a nymph, a major life history transition from aquatic to terrestrial insect, and then its ability to fly. And so, and we had the nymphs in all of the different treatments. So very excited to share that when it is ready, hopefully sometime pretty soon. But just kind of a bit of a spoiler of it, it's pretty challenging because we have 5 different traits we're considering, and the traits don't move in lockstep. So survival might say one thing, whereas growth rate says another thing. And so it's a pretty typical of multi-trait physiological studies, but it also highlights the complexity of really identifying, well, what is the thing that matters for this species going forward? And I will also link back to kind of the prevailing idea before any of these studies I've told you about was that Lednia humana and these high elevation stream insects are super sensitive to warming, and any amount of warming essentially imperils them. So the loss of the glacier directly imperils them because the water will get warmer where they live. And so our 2 recent studies suggest that there might be a little bit more optimism, at least from the abiotic standpoint, that as climate change proceeds and glaciers are lost, these organisms actually appear to be able to handle warmer temperatures or deglaciation, at least in the short term. But that says nothing about, what might happen downstream or what kind of biotic changes might occur with competition or something like that. But thinking purely physiology-wise, this experiment suggests the real answer might be somewhere in the middle. And so our optimum temperature for growth for across all of the treatments was something around 13 degrees Celsius. And so what that says is that, the optimum temperature, which there are challenges using that approach to determine optimum, but that temperature is much higher than they typically ever experience in the wild. And so that does indicate that there's something else going on with why they are in these such extreme distributions. And so with that, I want to talk just briefly for the last 10 minutes or so about the other work we're doing and how we want to integrate all of this into kind of a more robust future of working on alpine stream insects and their vulnerability under climate change. And so it's going to be long-term monitoring work, which Brielle talked about in the intro, identifying refugia, which links back to some of that subterranean ice discussion that I mentioned earlier, and then genomic resources and what that could mean for better understanding things like an RNA-seq experiment and better predicting evolutionary trajectories and understanding where they've come from. And so the first thing, I want to tell you all about the Teton Alpine Stream Research Project. This is part sharing it and part advertisement. If you're interested in working with us on this stuff or have an idea, we are all ears. It's a small, really powerful, fun group of people at a lot of different institutions. And we began this project in 2015. We monitor 12 sites in Grand Teton National Park annually. 2022 will be our 8th year of continuous data collection, which is insane to me, considering I was a third-year graduate student when we started this project. And we have a bunch of different hydrological sources. And the overarching question is, do rates of biological change in these streams depend on the source? And are some sources going to support a more stable long-term trajectory than others? We have a range of core biotic data, both microbial, invertebrate, eDNA, a lot of stuff. We're actually starting to work on some temporal genomics with the idea of seeing how individual species respond at the genomic level to loss of a glacier and the various selective pressures that come with that. We have a bunch of abiotic data we measure every year, including in situ temperature loggers. And I just want to give a shout out to a couple of the really great graduate students working on this project. Taylor Price is doing a really neat nutrient cycling microbial project looking at how the Alpine snowfields are linked into the actual stream ecosystem at University of Minnesota. And then Karen Jorgensen is going to have our first perspective on who eats what and who eats who in these Alpine streams. She's doing a really neat isotope food web study for her master's. And so this brings me to the rock glaciers and cold rocky landforms and the potential for climate refugia in these ecosystems. And so here are 5 different types of landforms that we call cold rocky landforms collectively. And these are all geological features that harbor ice or at least harbor cold conditions for downstream ecosystems, plant communities, whatever, in mountain habitats. The most well-known of these is the rock glacier. It is essentially a surface glacier that is covered in debris. So it still moves, but it's insulated from ambient temperatures and solar radiation. And they're really cool from a potential climate refugia standpoint because they're super common. So they outnumber glaciers and snowfields by 2 to 1 in the American West. There are something like 73,000 of them around the world. And we know from the geological literature that they are changing more slowly than surface glaciers due to that debris cover and insulation. And then from our work, we know that they support biotic communities that are representative of the mountain landscape they're in, and that include ESA-listed species like Lednia Zapata Glacier, et cetera. And so we think that they could be, as Ben Goldfarb, I got to give him a shout out. He wrote a really great book about beavers, which you should check out. Also a Washingtonian. He wrote a really nice popular press article about this idea from our paper earlier this year that these rock glaciers and related cold rocky landforms may act as refugia going forward because they may maintain stable cold conditions in a mountainous environment that's rapidly changing and warming around them. And so we're now working on actually integrating this into some conservation planning frameworks with the idea of actually laying out how do we monitor these cold rocky landforms and the refugia they might sustain and use them to actually assess species that are at risk. So we laid out some ideas for the Western Glacier stonefly, Zapata Glacier, pika, plants, terrestrial invertebrates, kind of thinking more broadly than just aquatic biodiversity to the full spectrum of biodiversity in mountain ecosystems. So that's really exciting stuff that I'm happy to talk more about anytime. And so I want to close with perhaps the least ecological directly part of this talk, but something that I think is really important and is obviously very quantitative, and that is building a genomic future for aquatic insect conservation broadly. And so this is work that I'm doing with Paul Franson at BYU and a few other great folks. And so just to give you an overview and some context, I'm not going to go into any deep in the weeds genomics here, but I'll just kind of lay out the motivation for why we're working on this. The first animal genome was sequenced in 1998, or was published in 1998 in the journal Nature, so about 23 years ago. And now these are all of the animal genomes. So this is essentially every species that has a genome assembly across the animal tree of life as of June 2021. And I don't expect you to be able to see everything in this plot, but I want to highlight that it's super diverse. So from bryozoa to jellyfish, humans, insects, mollusks. I mean, there are genome assemblies all over the place. There are about 3,300 total for 24 phyla. There are 10 entire phyla that still have no genome assembly. This still only represents about 0.2% of all biodiversity, but arthropods are massively underrepresented, including insects broadly. And so this column here, B, is genomic representation based on the number of species in a group. And what we would expect, like how many genome sequences would we expect given X number of species? And an asterisk indicates significant over or underrepresentation. Red is under, green is over. And as you can see, vertebrates, as we might expect, are massively overrepresented in terms of total species diversity for the number of genomic resources that there are. Whereas arthropods are massively underrepresented, except for things like Drosophila and mosquitoes and butterflies and those kind of groups that have gotten a lot of attention. And then what's really exciting about this also, this is on the y-axis here is a metric of genome assembly quality, contig in 50, which is how contiguous a genome assembly is. And on the x-axis is is time. And I just want to point out that as this technology called long read sequencing has emerged, we're getting some really, really good genome assemblies now. And so we're getting them faster and they're better than ever, but they don't include aquatic insects. So according to species diversity, we should have 9 terrestrial insect genomes sequenced for every aquatic insect. But instead, that ratio is 24 to 1. And when we look at quality, those aquatic insect genomes is the same metric on the y-axis. This is a bunch of different insect groups. Aquatic insect genomes are not particularly good. So here's 4 of the kind of primarily aquatic groups: mayflies right here, dragonflies, stoneflies, and caddisflies. But all of that is about to change because Paul and I and a bunch of others have recognized this, and we're developing some new resources, which we will share publicly, hopefully very soon as a data paper. And our big question will be, our first question that we're curious about is just the evolution of aquatic insects. And then once we understand more about where aquatic insects came from and how they keep evading fresh waters, then there will be questions about vulnerability to climate change and warming and other topics that are relevant to what I've just talked about. So some quick take homes in the last few minutes here. Climate change is dramatically altering our planet through glacier recession. We need a more thorough accounting of the biodiversity linked to glaciers, as well as the physiology and ecology in order to predict actual downstream vulnerabilities. It appears glacier-fed stream communities may be more resilient to climate change than we thought. I want to be careful there with some caveats, at least in the near term, and that really we need some long-term perspectives that integrate more biotic work, some species interactions and resource use into that discussion. There may be some really exciting future for identifying in situ climate refugia to monitor biodiversity and mountain ecosystems going forward, and that we need some more genomic resources to better understand the threats to these species for things like genetic diversity, et cetera. So with that, I will acknowledge some really great people that have worked on this with me and take any questions. So thanks. And I'll see if I can actually figure out how to get back to video and such.
Thank you so much, Scott. That was great. And oops, thought I unplugged myself. That was great. And it's awesome to be reminded how important aquatic insects are. So it's nice to, think about that again.
I'm happy to do that anytime.
Yeah. So as a reminder to everyone, you can put your questions into the questions bar and I can read them out. All right.
And if anyone wants to contact me later, doesn't want to ask a question in this setting, I totally get it. I often do not want to pipe up in seminars with my question. So feel free to send me an email or something. I'm happy to chat anytime.
You can also use the raise your hand feature as well if you have a question. Stephanie said that it was a great talk and it's fun to hear about your topics. So as we wait for maybe a question or two, I have a quick question that I was thinking about as you were giving your talk. So I'm wondering if there's any evidence or research that extreme like alpine events, like maybe like a wet avalanche or something like that is affecting biodiversity differently than the normal just receding, like how those two events are being impacted.
Yeah, so there's been a lot of like glacial outburst flood discussion lately. There was a really deadly glacial outburst flood in the Himalaya, I think like 6 or 8 months or a year ago. Actually, there's a really nice Nature paper about it now. We don't know. Yeah, so we haven't really, I don't know of anyone that's looked at the like biodiversity impacts of those kind of events. I would assume they're pretty major. But one thing I will point to is actually rockfall events onto glaciers cause the glaciers to stop receding as quickly. And so, because they add insulation to the ice. So you could actually make a case that for the long-term future of biodiversity, rockfall events could be a good thing, at least in the near term. But yeah, they're really dynamic places. And unfortunately, we just know so little about what actually happens in them. That was part of why we put a bunch of webcams out, or wildlife cams out on Mount Rainier, just to get a better sense of like, what is even up there? Like, what does day-to-day life look like when we can't be there? Like, and so I'd love to see more of that and more integration between like geologists and glaciologists that actually monitor glaciers, but don't think about, you know, biodiversity implications directly. So call for interdisciplinary research, which is probably a good thing in this crowd.
For sure. Yeah, we actually have a question by Karen, and Karen says, thanks, Scott, for the awesome presentation. Can you clarify whether the beta diversity metrics you presented were spatial beta diversity or temporal beta diversity?
They were spatial. Yeah, they were all spatial. Yeah, so actually temporal is something that we want to look at with our long-term data from the Teton range. We promised ourselves we would not start digging into it until we had 10 years of data in hand, because we don't think conclusions will be valuable until at least 10 years. But we've started looking, and we'll see what we find. But
cool. Karen says, thanks. Yeah, that seems like a cool, lots of data that you'll be sending out soon. So hopefully that'll be, fun to see.
There's a neat study from the Alps that actually showed that over a 10-year period, some of the alpine streams are warming by like a degree a decade, which is extremely fast. And so we're very curious if our data will bear out a similar pattern in the Rockies.
For sure.
It'd be really scary if they do. We don't think they will, but.
I'll just wait another minute and see if anyone else has any lingering questions. Again, super awesome talk. It's nice to hear about everyone's different study areas. So it's cool. So it seems like we don't have any more questions, but again, Scott is willing to answer anything via email or anything like that. So thanks again so much for talking today, and it was a great presentation, and thanks everyone else for tuning in, and hopefully we can see some of you next week for the seminar series again. So
thank you. Thanks for the invitation. I will try to find my way out of here.
All right. Bye.